This is the second part of the PowerPoint presentation. The first section we covered diffusion, facilitated diffusion, and active transport. I want to spend a little bit of time on osmosis, since osmosis deals with water. And if water is very important to life, we need to talk about it separately. Osmosis is still a form of diffusion. It moves from an area of higher concentration of water to lower concentration of water. So from high to low, just like diffusion. But again, we're focusing just on water. And we also need a semi-permeable or selectively permeable membrane. So if you take a look at this illustration down here at the bottom, this is called a U-tube. Let's not worry about these terms right here for right now. But the color blue here is water. Okay, this blue color is water. The purple circles might be, say, salt or sugar or proteins. And so if you notice on this U-tube, there's actually more blue color on the left-hand side than the right-hand side. So there's more water over here on the left-hand side. And so water is going to move from area of higher concentration to lower concentration. And so water is going to move to the right in this U-tube. When we're trying to figure out which direction the water is going to move or which direction osmosis is going to occur, we have to look at the solute concentrations. And the solute would be, again, like dissolved sugars, dissolved salts, proteins, amino acids, and so on. Our first type of solution is called hypertonic. That prefix hyper means higher than. And so we have more solutes. Say I dump a whole bunch of salt in some water. That'd be a very hypertonic solution. The second type is hypotonic, where we have less amount of solute, maybe like just a pinch of salt in some water. And then we have isotonic solutions, where we have equal amounts of solute and water both on the inside and outside of a cell. So in this illustration down here, over on the left-hand side, this is considered a hypotonic environment because, again, there's lots of blue. We have lots and lots of water over here on the left-hand side. And then over here on the right-hand side, not as much blue, so not as many water molecules. And so this will be hypertonic. Hypertonic, remember, is less amounts of water, more solutes. Over here is hypotonic because we have more water and less solutes. And then whenever osmosis takes place, we talk about how this is a net movement of water. If osmosis does not occur, there is no net movement of water. So an overall picture here, managing water balance. A cell's survival depends on balancing water uptake and loss. In other words, if a cell has to take up water, there's a certain way it has to do that to maintain balance, or if it has to get rid of water to maintain balance. And so again, here we have here's our hypotonic solution, our isotonic solution, and then our hypertonic solution. And we're going to take a look at each one of these in more detail. So the hypotonic solution, an example would be like fresh water or pure water. A hypertonic solution might be like salt water, and then isotonic is considered balanced. We don't have an excess amount of solutes or an excess amount of water. So the first one we're going to take a look at is hypotonic solutions. This might be a cell in fresh water. So fresh water be hypotonic. There's a very high concentration of water around the cell. And so over here in this picture, let's assume that this area here is water. We have an animal cell here on the top and a plant cell down here at the bottom. The problem with putting cells into a hypotonic environment is that the cell will gain water. Water likes to enter the cell because, again, there is a lot more water out here than there is inside the cell. And so water wants to go from greater to lesser. And so it will move into this cell. Okay, so I don't know why I can't seem to do my arrow there. Okay. Well, we got going on here with my pen. Same thing with the plant cell. Okay, that one seems to be working okay. Getting some glitches here. Water will move into the cell. With an animal cell, because we don't have a cell wall, it has the potential to burst open. And so that's this term lysed right here. There's our kaboom. An example might be a paramecium. A paramecium is a freshwater protist. And in order to survive in a freshwater environment, they have to have some way of pumping out that water that they tend to take up as they live in that environment. So what they use is called a contractile vacuole. And this pumps water out of the paramecium's body. And of course, it does require ATP. A pump requires energy in order for it to function. In plant cells, this is called turgid or turgor pressure. I'm going to write this down over here. I'm going to write turgor pressure. So when we talk about this, I'm going to say turgid, or it has more turgor pressure. And the nice thing about plant cells is this cell is not going to burst open because it has a protective cell wall. Let me go back to the animal cell up here. The potential for it to burst open, where's my pen at here? There he is. There is a term we write for this here. It is called, well, if I can find my, sorry, I'm having issues with my, there it is. Okay, cytolysis. All right, let me see if I can rearrange my pen here. Where's it at? There it is. Okay, I'm going to write it up here. CYTO, cytolysis. Cell lysis, cytolysis, basically cytolysis. So the cell is going to burst open. So make sure you write that down in your notes, cytolysis. And then the plant cell down there would be turgor pressure or turgid. So here's a picture of a paramecium. Here is this contractile vacuole, and it will pump out. So here we see a picture of the contractile vacuole that is squeezing itself. So it's going to remove that excess water out of the paramecium's body. Here's our ATP being used. The second solution is hypertonic solution. This would be like an animal cell in salt water. Salt water would be hypertonic because there's more salt than there is water. Has a very low concentration of water around the cell compared to the amount of salt. The problem with this one is the cell loses water and they can potentially die. And so what tends to happen here is water is going to leave the cell and the animal cell is going to start to shrivel up. An example would be any type of shellfish. We'll talk more about this when we get done with some of these assignments, looking at these three solutions. But animals that live in salt water, they are prone to losing water from their cells because they're living in a hypertonic solution. And so they tend to have to either drink more water or find a way to pump out the excess salt. In plant cells, again, water is going to want to leave the plant cell. You can kind of see where it's pulling away from the cell wall. And so we call that plasmolysis when the plant cell starts to wilt. That is plasmolysis. If I go back up here to the animal cell, there's another term here that we can use called crenate. CRENATE. That's when an animal cell will start to shrivel up. Crenate or crenation. With plant cells, plasmolysis, they can recover if you don't wait too long to water them when they've been wilted. And then the third one is isotonic solution. Animal cell immersed in a mild salt solution. In fact, your blood is approximately 0.5% salt, so not a very big concentration there, and so you don't lose a lot of water here. Water goes in and out kind of at an equal rate. So there's no difference in the concentration of water between the cell and the environment. So animal cells really like being exposed to an isotonic solution. Now, one thing you need to remember is, like, if you don't have any water moving out, like in a hypotonic or hypertonic solution, we say that there's no net movement occurring here. So basically, you have the same number of water molecules going in and out of the cell at equal rates, but it's a very, very small rate. And so we say that there is no net movement of water. The cell is in equilibrium, and the volume of the cell is stable. Example would be blood cells. I just mentioned that up here about your 0.5% salt solution. If you've ever gone into the hospital and had an IV put in, they will put a saline solution IV hooked up to you, and it has to be about the same percentage as your blood, otherwise we have issues. Now for a plant cell, let's see if I can bring this down here. A plant cell says it's called being flaccid. I'm sorry, let me see if I can work through this here. Down there it says flaccid, so it's not really nice and turgid, firm, but it's not wilted either, so it's kind of in between. There were some scientists that started studying transport proteins back in 1991, and they were given the Nobel Prize in 2003. And what they discovered is that there is a type of transport protein called an aquaporin. And this is designed specifically just for water molecules. And these aquaporins will move water very rapidly into and out of cells. And so this was one of the first evidence that there were actually water channels within the cell membranes. So again, these are protein channels that allow flow of water across the cell membrane. And this is a computer-generated image of those channel proteins. And the two gentlemen that received the Nobel Prize was Peter Agre and Roderick McKinnon. So now, just a quick brief understanding here, or review. So we have a cell here, and inside the cell, it's 0.05 molars of solution. It says we have some sucrose in there, we got some glucose in there, and then out here in the water, it's 0.03 molarity. So we have some sucrose, some glucose, and some fructose. So what we want to do is the cell itself, this right here, since it has a higher number of solutes, we can say that the cell is hypertonic. And then the environment, the beaker, has a lower amount of solutes, and so we say that is hypotonic. And so water is going to go from where there's more water to where there's not so much water, and so it's going to move into the cell.